Welcome to Red in Motion from the American Journal of Respiratory Cell and Molecular Biology. What cell types come to mind when you hear asthma? The eosinophil, or perhaps the neutrophil, a cell that infiltrates the airways of nearly all asthma patients, but whose role in asthma pathogenesis and airways remodeling remains obscure. Vargas and colleagues sought to shed light on the intercellular communication of neutrophils and airway smooth muscle cells in their publication, Neutrophil-Derived Exosomes, a new mechanism contributing to airway smooth muscle remodeling, which appears in the September 2016 issue of the American Journal of Respiratory Cell and Molecular Biology. The investigators hypothesized that neutrophil-derived bioactive exosomes signal airway smooth muscle cells to proliferate. Exosomes are nanovesicles rich in proteins, lipids, and multiple RNA species that play a well-known role in intercellular communication. So, this study aimed to define if neutrophils release exosomes at all, and if so, what is their proteomic and functional profile with regards to influencing airway smooth muscle? Because of the need to obtain large blood volumes, the authors phlebotomized horses to obtain peripheral blood neutrophils. A sophisticated centrifugation, filtration, and size exclusion protocol permitted isolation and purification of nanovesicles from these neutrophils. The resulting nanovesicles then underwent examination with electron microscopy, size measurement, and proteomic analysis. LPS treatment of neutrophils in exosome co-culture with airway smooth muscle cells then provided a functional readout for the study. The data showed that neutrophils release exosomes displaying an expected profile of size, morphology, and surface markers. Within exosomes derived from unstimulated and LPS-treated neutrophils, the authors identified over 270 proteins, with around 5% being differentially expressed with LPS treatment. Functional enrichment analysis determined that the differentially expressed proteins regulate immune responses and intercellular communication. In co-culture, airway smooth muscle cells rapidly internalized neutrophil-derived exosomes and augmented their proliferation, particularly with exosomes derived from LPS-treated neutrophils. These elegant studies show that neutrophils release bioactive exosomes and that, upon uptake, they stimulate airway smooth muscle cells to proliferate. These findings carry important implications for the study of asthma pathogenesis, airways remodeling, and neutrophil cell biology in general. Thanks for watching this video highlight from Red in Motion. For more information, Vargas and colleagues' article can be found in the September 2016 issue of the American Journal of Respiratory Cell and Molecular Biology.